Okay, the prediction of protein-protein interactions, definition, protein interactions, what I'm talking about is transient. What you see here is a complex of the glycoprotein 120 from HIV in green, complex with CD4 and 2 antibodies. The structure has been done by the lab of Wayne Hendrickson here. And in fact, it was one of those really complicated structures to get, very important structures. Now, for our definition for this lecture, the ones that touch are considered interactions, and the ones that are going off and on are transient. So this one here, the yellow, the parts of CD4 that bind each other is not transient. So they're always in connection, so this is a domain contact. Again, the red ones here are transient. When I have a situation such as this one, where A activates B, activates C, activates D, then what I'm talking about is associations. So A, in particular A and D are associated because they're part of a pathway, they're part of a process, but they are not in connection, in physical connection, just as much as antibody 2 is not in connection with CD4. Okay? The standard way in which you measure high throughput protein interactions is used to hybrid, or one of the standard ways in which you want to establish that X and Y interact. You put 2 parts of a transcription factor, the binding domain and the activation domain. Activation domain you put onto Y, binding domain onto X. And then essentially what happens that if X and Y bind, then there is some upstream specific activation event that emits light, so you actually see a signal. That's the most common way for a high throughput experiment. There's mass spec, but mass spec tends to really bring large complexes. So mass spec sees everything that sort of sticks together, while these 2 hybrids really allow you in a high throughput manner to identify particularly pairs of proteins that really interact transiently and physically. Now, the first issue always is homology-based inference. So I want to predict that 2 proteins interact. The first question is, where can I do with homology-based inference? So now it's the same as always, just in this particular case, through the pair situation, slightly more complicated. So you know that A and B interact. You know that experimentally, and you have a protein A prime that is similar to A, and a protein B prime that is similar to B. I want to simply say that A prime and B prime interact. So the reason why this is more complicated is because now, always so far in localization and whatever we did so far, we always asked what's the similarity between the protein and its homolog. In this particular case, now we have 2 similarities here. We have the one between A and A prime, and we have the one between B and B prime. And now you have to make a decision. So are you going to look at whatever is minimal between them? Or are you looking at the max between this and that? Or are you going to look at an average? So there are different models. And what I'm going to show you in the following is where we look at the minimal. So we ask, so if say A is more similar to A prime and A are more similar to each other than B and B prime. So in that particular case, the deciding factor, the more diverging pair would be B and B prime, and that would count. And that's the way I'm going to show you the next curve. Well, in some sense, we can clearly use this, because this is, if we couldn't infer that A prime and B prime also interact, that would be the end of all sort of model organisms, right? If the interaction is only valid in human, and if the mouse interaction would be completely different, there is no point in using model organisms. We have to understand that. So clearly it has to happen, and it has to be able to, you have to be able to do it. Okay, let's look at the results. What I show here is the HSSP distance Let's get back first. In this particular case here, A and B were taken from warm, meaning I have experimental information for A and B in warm, and then I sort of try to predict, in this particular case, I also have experimental information for A prime and B prime that happens to be, in this case, in human. Actually, on this plot here, it's everything else, not only human. It's A, B prime, warm, C elegans, that is, warm, and the other one is anything that is not in the worm, human and others included. So for increasing similarity, and that brings us to the point of the HSP value again, the HSP H-val 0 is here, the 0 point is here, the values 30 would be somewhere here, the last one here is 25, so that is this value. Now, what you observe, and again, the points, the green points, are the data. The axis here, let's just ignore. But the higher the number, the more they agree. Whether that is 2% or 2, let's not get into that detail. But the first, and the red one is the error estimate. Okay? So my first question to you, can you see that homology-based inference is better for higher sequence similarity? So can you see that this curve goes like this?
With some intervals.
You cannot clearly see that, right? It is, you would argue that you can clearly see it's not like this. You can, so if you had to compare this versus that, this is clearly less likely. But whether it's straight or like that is not clearly distinguished, right? If with some goodwill and looking at the issue here at the first points, where in fact the error estimates are much lower, then you do see that there's an increase. Then the error rate gets so high here that you cannot really continue that curve. So this is the way you would describe it in your results section in your thesis, that where the error is low, where you can trust the data because you have enough data, you at least see a tendency of a clear increase. From here to here is a clear increase. Even within the, well, not quite within the error limit, but almost exactly. At least in the beginning, this story looks okay. Either way, so there appears to be some tendency that you can do it, and there's some tendency that you can do it better for more similarity. Now, one thing is clear. The story that we had here for subcellular localization in blue, for enzyme in red, so that's all 4 digits of the enzymatic activity, is at least the first digit, so it's a lyase or something like that. You see that there is a clear function, so the more the HSP distance goes down, something falls and then there is a phase transition. There's nothing like that to be seen. Error or not. It's clearly not as simple as that. That is also clear. Okay, let's get into the next issue. Typically, what we have is we assume that there's a corresponding pairs in genes of different species. What about lateral gene transfer? So ultimately, what we're talking about is, again, the origin of the word homology is we have an organ in whale that is similar to the organ in human. It doesn't quite do that same function, but it does something. So when we swim, the hand here is still sort of doing something similar to what the whale does with the hand. No, it's not true. Does anybody... how is this for the whale? Which one is the hand and which one are the legs? Who remembers this? It's a little bit more complicated. I can't remember whether it's the feet or the hand. And the fin, is that the...
Of course, the feet in dolphins and whales are not really there.
Yes, they are. They are. So So what is the fin? Which one is the fin?
I think the back.
No, but is the leg, is one of the legs, or so it's not like
No, the legs just don't. I don't think they go outside of the wheel.
I thought it was.
Pretty sure.
Okay, we have to discuss.
Outside.
What did you say?
They're just inside.
So that's what she
I
said.
would say they are just sort of remaining bits of bones on the inside close to the vertebra.
Okay, so I got that wrong. Anyway, so then that's the hand, and that is actually doing the same thing when I swim. So completely homolog. Now, the issue in genome evolution is, so we start from species A, we sort of split between two species, and at the first moment of this species split, you essentially have the same genome, right? Or very, very, very similar genomes. And very similar genomes here, what I'm indicating in terms of 3 proteins make up the genome, say. And these three proteins or 3 genes here, they are the corresponding ones. And B1, this one, and B2, this one. And they do the corresponding things. And then there's another sort of speciation event, and they are still similar. And then here's some duplication event. In this duplication event, the idea is that we create something new, because the original 3 that we needed for some original function are already there. So we have the freedom by the duplication event to create something new. That's the idea, right? And that brings us to this term of orthologs and paralogs, where the orthologs are the ones where essentially we have the corresponding. So this is the ortholog of that one, that is the ortholog of that one. And this one here would be the paralog. This is the one that happened through a duplication event that was free to evolve a new function. That's the idea. Okay? Now, that brings us to this issue of duplication is one way of doing it. Another one is to jump in the gene, right? So you have, as you all know, there are these situations where you put a gene on a table, and then you look at it, and then it jumps. Right? Do jumping genes exist? They do. Very few people do it on the table, but they exist a lot in plants. So this is why people explain the different color of corn. When you look at the corn cob, sometimes they have different color. So not the variety that you buy here, but when you buy in New York, you get typically the corn that you buy to eat has many different colors, the cob. And that comes essentially from jumping genes. Here's an example for the sea slug that in fact incorporates chloroplasts from algae. And this is essentially kleptoplasty. You get an idea, so this is sort of stolen, but essentially, there are jumping genes. There is all kinds of processes of horizontal gene transfer in bacteria. So there's a lot of other than duplication events that create paralogs or create things. Now, this also makes it more complicated because a jumping gene now can be a paralog and ortholog. It can be that the ortholog jumps in there by coincidence, and that makes the whole story much more complicated. That brings us to this issue now. This issue being that, okay, now this is the example of what I talked about before. Let's make it slightly more complicated. Now we have a protein A prime, and A2 prime. And the A2 prime and B2 prime are in a different organism. A prime and B prime are in the same organism. So typically what we would have here is here we would talk about A and A prime being paralogs, and A and A2 prime being orthologs. That's typically the way the model would assume it, right? And now let's assume that we have cases where A and A prime is as sequence similar to each other as A and A2 prime. We still would typically call this the paralog, because that is the duplication of jumping gene or kind event in the same organism. So now the question to you, I'm going to show you the data now that I had before. That is what I showed you before. And so the first question to you, is that now this one or is it this one that I show you? And where would the other one be? So I'm going to show you 2 graphs, where one, the pairs that I show you, this one is warm versus not warm, so this is essentially this and that. A2 prime and B2 prime compared to A and B. My question to you now is, where, on the next slide, I'm going to show you A prime and B prime, the paralogs. So what I show you here is the orthologs. Where will the paralogs be? Okay, there's a lot of error, but tendency. Same place? Below?
Yeah, well, the paralogs are supposedly below in similarity.
So we will somehow drop, and then we have some issue with the error rate, so it's unclear how much we will drop. But this is what we expect. Anybody has an issue with that? And of course, this is exactly what we observe. This being biology, right? So as you see, the curve is there. Why? Absolutely no answer. When the problem when you discover things like that, this is the kind of thing that is very, very difficult to publish, because nobody expects it, so nobody believes you. And then they make you repeat and repeat and repeat. So here is the same story for fruit fly versus fruit fly. So orthologs here. The orthologs for fruit fly versus everything else, and then the paralogs within fruit fly here. Now, in this particular case, we don't really have a label of paralogs, orthologs, because of a variety of reasons. But what you do see is that wherever you put your threshold, the P story, the within the organism, is always higher. And that's somehow true here too, although there is no point up there. But even within these high error rates, it's clear that there is a difference between the blue and the green. So no matter how big the error rate is here, actually the blue points are still staying away from the green ones, right? Now, it remains unclear what really the reason is. Predrag Gradivojac has observed something like this also for enzymatic activity and for Go numbers. So he essentially asked, how well do you do in homology-based inference for Go numbers? And he published a paper that showed that in fact the inference of Go numbers also works better for Indiana University, works also better for paralogs than for orthologs. And again, why is unclear. At this point, it's a very clear observation. Others have sort of repeated this. Paralogs conserve interactions, orthologs don't. Seems to be the case. And clearly this means that there are some issues of using model organisms. So this means that what is exactly not conserved is what we typically try to do with a model organism. We try to infer the connectivity. We try to infer the pathway. We try to infer that if I have a disease in human, and if I put the same disease gene into the mouse pathway, then the mouse would somehow evolve a disease. If the networks are not the same, this is highly dubious. And to some extent, that really is an issue. That brings me to another question. The question of what happens if I measure the same interaction twice? In this particular case now, I use AB. It's not a homolog. So I measure A and B twice. Okay? So this is not the homology-based inference here, where I have some sequence similarity between A and A prime. No, now we really ask the interface between A and B in one experiment, and the interface between A and B in another experiment. Somehow the same conditions, but let's just compare two different X-ray structures. Let's compare two different experiments or experimental labs determining the interface between two proteins. Will they agree? So you have all heard about this lock-key idea, that the way two proteins interact is like a key and a hole. So they recognize one particular conformation, and it fits only in that conformation. No other conformation will fit these two proteins, right? If that were completely true throughout, then you will always observe exactly the same interface. Now, how do you measure interface? What would you do?
Fred? First resonance?
No, I mean, sorry. What we have here, you take what you have is two different X-ray structures of A and B in interaction. Okay? So now you all do it in the computer. And in this computer, you're asking, are these two interfaces the same? I have one solution from one lab and another solution from another lab, or slightly different conditions or something like that, right? My question to you is, how do I define interface? Or when do I say the two interfaces? How do I measure whether two interfaces are similar? What do I measure?
Cartesian distance between the atoms.
So that means I look at the sum over all atoms in the interface?
No.
So what?
Depending how we define interface and all atoms, so we can take all the structure. We just take the one that are closer than some cutoff.
Good. We take some threshold. Say we say closest atom is less than 4 angstrom. And then I say they are connecting. Okay? So if we have two residues of two different proteins, and if the closest atom is at least within 4 angstrom, then I say they are in contact. Okay? Now, this still doesn't define my interface. Or it does define my interface. So now I know all the residues between protein A and B that interact. And now my question is, how am I going to look now differentially at these two interfaces?
That's not what I meant when I said distance between Cartesian. We should align the structures.
Yes.
And then we'll have...
Wait a minute, align the structures, but we have the same sequence. A and B are A and B. Should I really align the structures?
Definitely, because there is no guarantee that at the moment that the X-ray was taken that they are actually identical. We would need to check that.
The sequences are identical? What do you mean?
The sequences are identical, but it doesn't mean that the complex will have the same 3D configuration.
It will not. It will not have the same 3D
So that's what we have to measure. And then the deviation between every...
So what do you align? The pair? Or you align each structure?
Structure, and then we need to measure...
Each one of them. A and B? Or what exactly? The complex?
We align AB to AB, and then we see the deviation for each pair of atoms that should correspond. So we'll have in protein 1, we'll have atom 1, which is present 2 times. And then we need the Cartesian coordinates.
But again, so I asked you a question and you didn't answer that. That brings me ultimately to the point that where, for instance, say there are 20 residues in the interface 1, and then I will do the RMSD over 20, or per residue RMSD, or what do you look at?
We would need a measure for that, but I think there's some publication.
That's what I'm asking for.
I think Manfred Zippel did something in that direction.
That is true. Yes, that is true. That is very. But so the issue, Manfred Zippel does not quite do this. Manfred Zippel asks, when I look at Manfred Zippel answers exactly your first point. I want to align this entire complex here with this entire complex here. And how much do they differ? That's what he has the measure for. And that's what he has the method for. I'm only looking at a tiny fraction of this complex, which is the binding site. And his measure is may not quite cover this.
So we can boil it down to that.
Not quite. Because it's an overall difference. We need something that is very local. Anyway, how could you do it?
Look for mutual exclusivity between your measured contact residues in contact. So you can define different interfaces if they're mutually exclusive. Like you get
You lost me.
You get a number of probabilities, like if you measure the same interface or the same protein interaction like 20 times,
Yeah.
you get a distribution for the residues in contact.
Yes, that's exactly what. Let's look at two times only. This is exactly what I'm asking. A distribution of the residues in contact. How do you measure the residue? What do you mean by that exactly?
Like we have defined a measure of one residues are in contact. Like
Yes. And then?
and then we get a list of residues in contact.
So essentially, what your percentage of residues of contacts
Shared, I guess.
shared.
If we only have two.
Shared. So now shared. So I have residue 42 and 69, and 42 and 69 in contact. Yeah. That is what we would call a shared one.
Yeah, that would be perfect.
Okay, that is one solution. Can you think about another solution?
Oh, if we have all the data, we can look, of course, if they're in the space nearby located.
So this would be slightly different. So there I would ask, let's call that 1, 2, in same bubble.
So we can.
So what I define around the binding site, I define a bubble, and I ask, are all the ones that I contact here around some within that bubble, and vice versa, right? That's another way of doing it. What else could I do? Same residues. So I can simply ask, okay, this definition here of the 4 angstrom defines me a set of pairs, and in interface 1, the set of pairs involves 20 residues. Maybe the pairing in the second interface is different, but are the same 20 residues involved? Which is not quite the contacts here, right? It's sort of more loose. I would allow to sort of have some local arrangement, but at least I ask, are these involving the same residues, right? What else could you look at? Essentially, you can ask, is it, so the contact formation leads to the exclusion of water. So is the same thing excluding water? So are you excluding water in the same way? So is somehow even if the pairing is not the same, even if it's not the same residues, but is the shape of the interface essentially similar? So you're sort of excluding the same amount of surface. And these are 4, I believe I can't remember what else we tried. I believe we came up with 6 different ways of measuring different interfaces. And then we asked, how many of the proteins are in fact agreeing by all these, are at least agreeing in one. This is what we're looking at. So for 90%, in one of these 6 measures, they agree. On the other end, for about 10%, they disagree by all 6 measures. So we have some fraction where we clearly can say these interfaces differ. We have some fraction where at least there's something very similar. But even that is not 100%, that's 80%. Here, the number. So in every single case, we saw some difference. So this idea that we essentially could look at the entire same contacts, that we sort of can align those things in the binding site, was not valid for any of them. I mean, not any is wrong. We had thousands of pairs here, but on average, at least, it was not. Here are 3 examples where, in fact, these 2 here, the blue and the orange, are different solutions. It's the same binding, and there's no matter how you measure this, this is very different from that solution. Here. Again, these 2 different interfaces do differ, not as much as here. Here it's here and here. those are two different binding sites. And again, this is very, very, very clear, right? You bind on different sides of the molecule. And both are biological interfaces. So we were surprised to find that. So it is seemingly true that you can form a pair of protein AB with very different types of binding modi. And again, both of those solutions here are biological. How common is this? Again, this work suggests that it is quite common. That these are extreme examples. But it's quite common that you have a flexibility that may be higher than what we clearly expected. Okay, so let's get back into, can we predict protein-protein interactions from sequence alone? The first way I did it is I essentially looked at a window of 9 consecutive residues in protein 1, 9 consecutive residues in protein 2, and I tried to predict at the center, in here it's 5, so whether the central of the 5 is in contact. And then I simply was sliding this window through the 2 proteins, right? So here, this one came from the first to the last residue, and this one came from the first to the last residue, and I simply tried all possible combinations. Do you believe this works? Highly not. This was by far the worst failure I've ever achieved. The best failure, let's call it that way. So the first, ultimately the problem was I had too many false positives. Now too many false positives could be because when I slide all of these 9 mers through my protein, I have many 9 mers that will never meet each other because it's not on the surface. Maybe they would bind, but since that 9 mer is inside of the protein, it cannot bind another protein, right? So it's a false positives that will not happen in nature. But so the first way to correct for that is I predict solvent accessibility. So essentially I distinguish between the core here and the outside. And in this image here, let's say 50% of the residues are on the surface. And by predicting this 50% on the surface, I can reduce the false positives by a factor of 4, as 1/4. Because I now only have to look on both sides at the 50% on the surface, which is 1/4 of all the possible sliding 9 mers, right? Then, in fact, I can go one further and I can predict residues in external interfaces. So I can essentially predict not only that this is the surface, but maybe I can predict the residues that bind to other proteins. And I would immediately, if I could, I would have much fewer windows to slide. And I would throw away a lot of the false positives. Every single time I throw away false positives, I also throw away some true positives. But most of what I throw away are false positives. And I will talk today mostly about this point, but let's just continue on the slide here. The next one then would be to predict residues that are saturated internally. That sort of is the flip side of that coin here. I predict subcellular localization so this shows how proteins, what's the probability of proteins in two subcellular classes, compartments interacting with each other. Green means more than expected. Red means much less than expected. Now if I simply turn this around and I said I will only consider the diagonal. So these ones here I would completely miss because I will throw them away. Here are interactions that are important, but I throw them away simply because I only consider the diagonal. But still, in a situation where I have 6 different states, I throw away 36 times of I reduce my false positives by a factor of 36, right? If they would be equally populated. And as you remember, log tree 2 predicts subcellular localization in 18 classes, so it's 18 squared. The reduction of false positives is a big number. Again, I throw away true ones too, but most of what I throw away is false positives. Now, then we can predict protein in substrate interfaces. We can define exactly where the domain is and improve alignments, and we can put all of that together and predict binding sites. And Tobias did his PhD on this, and I will get into that most likely either at the end of Thursday or next year. But for the time being, let's begin with the data collection for the protein interaction residues. So the first question that I have is, so we have again the GP120 bound to the antibody in the CD4. The first question is, these different types of interfaces that I show here, the red, so this is external, the green internal interaction, and the blue here is between dimers of the same protein or something like that, right? So my first question to you is, are they using the same biophysics? Right? And that is an important question, because when I collect my data, I take all of the PDB, but then the next question is, what type of interfaces am I going to look at? By the way, there's another issue here, how to extract the interactions. The first one is, of course, I had to take a sequence unique issue, but that brings me back to all structures, but that also brings me back, how do I define contact? So typically the way we would do that is, so that's the GP120, that's the CD4, so we would measure exactly the way I said it. We would have a distance between the closest points between these two, and then I would apply some threshold, whether or not that is closer than some threshold. I'll get back to that in a moment. But there's another issue. The other issue is, chains have breaks. When you look at the PDB, you will see that there is no simple one sequence, and that is one protein. You will not be able to see from a PDB chain, you see chains in the PDB, and there's chain A, B, C, D. How do you find out whether A and B are coming from the same protein or from two different proteins? You may want to find some header that says, yeah?
I'm pretty sure that if it's from, no wait, with different proteins, you don't mean invisible, just you mean different chains.
So I really mean, so in PDB you have so you have a protein that is one PDB, and one PDB has chain A and chain B. And chain A and chain B have residues that are close to each other. So it's an interface. Okay? My question is this an interface between two different proteins? Or are these two chains in the same protein? How do you find that out?
Read the description and hope it's annotated.
Yes. This is in fact how many publications did it. And you're going to do that for 5,000 proteins or 10,000 proteins?
Probably not, no.
So what do you do then? And in fact, by the way, that's the reason why, I'll get back to that in a moment, why many publications, in fact, were basing their answers on 50 proteins.
Yeah.
Because there's only so much that we can really carefully do.
Either fill out those with breaks or separate chains, or I don't know, train a machine learning approach.
But for that you need training data. And you're 50, so then you have to do it for 1,000, because otherwise you don't have enough labeled data for training. So that's not going to do it.
Use a very stratified statistics approach to see whether they are in the same category or not.
But how? How do you find that out?
That's a good question. Yeah, like, I mean, you could see whether whether the chain starts with a methionine, I guess.
So in PDB, most chains do not start with a methionine.
Yeah.
And the next issue is you find the next methionine, and then you will whether the chain starts with methionine. No, in the chains, no. Not going to work. Interesting idea. Very interesting idea. In fact, many, even in Swissport, many. So there's the first variant, sickle cell anemia, is E6V. So that is the first variant that ever where people showed that a particular sequence variant in fact implies a disease, sickle cell anemia. And the way you typically read this here is the E at position 6 is mutated to a V. It's actually at position 7. And that's because of the methionine being cut. And because of the structural biologists, so that was discovered by structural biologists, and they don't see the methionine. So unfortunately the methionine is not going to help here. Yeah?
You could just download a protein sequence database like UniProt, then extract the sequence from the PDB structure and just do a string search through the database. You find the identifier in which codon the chain occurs.
Yes, there is a tiny bit of an issue here. UniProt is populated essentially automatically. So how do you know whether the two different identifiers come from two different transcripts or from two different proteins?
Are the transcripts mapped to a protein again?
In some cases, yes, but not on the level of... So if you talk 100 million sequences, which is essentially what you mean by UniProt, then you cannot rely on this statement being correct. Yes, for human they are. So you could do a subset of human. Okay, that would be one solution. But is there another way of doing it? You're very close. So if you did the same thing with SwissProt, at least in SwissProt, you have an experimentalist who actually used that protein, that chain, and showed that this is a protein. Still not quite, the transcripts are mapped back, but typically the entry into SwissProt requires that somebody did an experiment on that protein. So there is some protein out there. So the probability, if you look at the subset of SwissProt, the probability that there is some fishy things is much less, and then this is exactly your idea, Gregor. Yes, and that could, by the way, I never thought about, now we could go back and sort of try to learn, but I don't believe this is easy to learn. I'm not sure. But anyway, so this is the way you can create a relatively large data set. And then you can look at certain things. For instance, that graph was surprising to me for two different thresholds in terms of what you consider a contact. Either the closest atom being 6 angstrom in blue, or the closest atom being 4 angstrom in yellow. What I show here is how many residues are in connection in 9 sequence consecutive. So you cut 9 consecutive sequence residues out of the interface, and you ask if the central one is in the center one of these 9 mir is in a protein interface, what's the probability that the other around it are also in an interface? And what you see here is that for the blue we have a peak around 6. So for a 9 mir, 70% of all the residues in a 9 mir roughly are also in the interface at a definition of 6 angstrom. When you go to the yellow, you sort of move a little bit to 5, 4. But what you see here is that the protein interactions are formed by things that you somehow can see on sequence. This is not like enzymes. Histidine triade in an enzyme is the definition where you really have 3 histidines that come together from very different places in the sequence, and they come together and form the binding site like this, right? And these 3 isolated histidines are not close in space. So if we did that for the histidine, then the max would be at the 1. It would be only the histidine, and then in the entire window of 9 there would be only histidine binding the substrate. And then when you get to the next histidine, this is way more than 9 residues away in sequence, right? So the enzyme story would look like this, or much sharper actually, most likely. So we were very surprised and very encouraged also that we see that, because it implies that if you somehow want to predict from sequence, there's a good reason to hope that you can have a signal. Because it's something, when you look at a local neighborhood, you have the signal. It's not only 1 residue. In fact, most of the cases, more than 1 residue in that. Now, there is another issue. And that is issue, unfortunately, I'm detouring again. And I go into a method that predicts flexibility. So you have seen NMR structures, you have seen this could also be different crystal structures of the same protein overlaid. So this is hen-wide lysosome structures, and typically where you see everything on the same, where it's very, very dark and well focused, is very rigid, meaning that all the models exactly find that solution. While where you sort of were frizzing out here, where these arrows here,
I thought it looks cool.
So just the shape. This is actually not hand-wide. It's art. It's just an artist who drew that. Anyway, so the mobility or the flexibility is in these regions here, right? And our hope was we can predict it. If we want to predict it, first there's something called the B-value that sort of reflects this flexibility, the mobility, the degree to which you can move, the functional size. And when you plot that for a large set of structures, so depending on the resolution here, there are thank you for interrupting. And then, well, let's get into that. Anyway, so there is an issue about the threshold. In this particular case, we chose a threshold that is far away from the maximum of the distribution. Here we choose 6 angstrom closest atom. And then we ask the questions, do these different types of interfaces differ? So if that is your question, and the question is important because I want to predict protein interfaces, if I could take if the biophysics for internal binding and external binding would be the same, I would have a lot of data to train from. Of the red types of interfaces, I have much fewer in the PDB than of the green types of interfaces, right? So the question is it the same biophysics or a different one? How would you answer that question?
It's different simply because they are by force located in the same space, so they arrange the best they can instead of...
This is an impeccable argument. Essentially what you say is they are of course different because I believe they are different.
Yeah.
The problem is, it's like religion, right? You cannot falsify that statement. I believe in God because God creates everything.
I could choose to verify my assumption.
How would we verify that?
Yeah, because we have the data, I could see if the...
It's true. I take your point. My question to you, if I asked you to answer the question today, you don't have the data, so what would you do?
Not different. Not different. Not different physics.
How would you arrive at so you say the opposite, just for the fun of it. This is a valid process, but typically what would you do? You would Google, right? And then you would find things and you would see that some people publish and say they are different, and some say they are the same. Or the other way around. So what would you do next? So I give you, I believe we had in the ballpark of at least 6 publications, 3 on either side of the spectrum. 3 said they are different, and 3 said they are the same. So what would you do next?
Train one system.
Before you do that,
what is the formula and hypothesis on the basis of which we could then decide what would be acceptable as a measure of...
We're talking today. We're not talking about I have the data and I can do something. We're talking today. What would you do next? I give you 6 papers. And 3 on one side and 3 on the other side. What would you do next? So we would read the papers and see whether there is something in the method that doesn't make sense to you. And in this particular case of the 6, what we found out, and this again is not untypical, what we found out is that we had issues with all 6. So that didn't do it. Okay, then at some point we're going to, on the next slide, we're going to go to the stage where we redo it ourselves, we have the data. But that took us actually, to get those data, took us a year. To come up with a Swiss plot idea, in fact, took a long time to set that up. It sounds very trivial, but it was not as trivial as it sounds. Now, the difference really between the method was also that people who simply had followed the assumption that they in fact are different, they had formulated a hypothesis that the biophysics is the same, and then they had looked at the entire switchboard, the entire PDB. And others, so that's a large data set. Others had really carefully looking at the interface and deciding whether that really is a different protein, what you initially said, but they could do that only for 20 proteins or 30 proteins. So this was difficult to compare. So an analysis on 1,000 proteins and one on 20 proteins. Anyway, so the next part of the algorithm that you would do is, what would you do next? Still in those papers. You would ask whether you know them. And that again, I keep telling you, networking is important. You need to, when you present papers from people, both in your seminar and also in your next group where you do your thesis, present clearly the group that did it. Because there may be people in the room, you have to remember who are the main players in this field. That's part of networking, right? And you have to remember the people you can trust. Science is also trust. Don't underestimate that. And if you don't know anybody, what still would you look at this paper? There's something else that induces trust typically. What is it?
H value.
Yeah, well, the H value is one way, but even before that you look at the journal. So if the ones that say it's the different biophysics are all published in higher journals than the ones that say it's not. If the place where they come from, if it's all Ivy League schools or compared to non-Ivy League schools on the other side, that may be. If all people who say it differs or doesn't differ come from tiny space on the globe, and everybody else is spread. There are a couple of things you can do within 10 minutes or 15 minutes that do not require to look at the data. So long before the year is over of getting the data, unfortunately in our case, so there were many friends on both sides of the fence for this. So that ultimately for us was the killer. So we felt they were all doing something wrong, but there were people we liked on both sides. So there's only one way we have to redo it, and that is what we did. And that's what we got. Now again, what I'm showing here is the difference with respect to all residues. Okay? Less than expected, minus values. More than expected, plus values. 20 amino acids. Internal green. The external, that's the transient protein-protein interaction in red. And sort of the oligomers, so the part of different chains of the same protein. Homodimers and things like that. Okay, and what I now like to do is assume you get in your thesis to the point where you have results or in your paper. Then all you do is you describe what you see. No interpretation. And I'd like that everybody picks a small part from this graph and describe what they see. Not the big story is, something smaller, so that everybody gets a little part of the pie. But go ahead, who wants to go first? Observe something in that plot.
I have a question.
Fair enough.
Yeah. Why are the residues grouped the way they are grouped?
So this is an attempt to make some to sort of, it's not alphabetical because somehow some residues are more related to each other than others. Okay, so positive charges, negative charges. It's a little bit more complicated than this one here. Polar residues or whatever you say. These are hydrophobic residues. We get into the functional residues here and other residues in some sense. The small ones and proline in there in many ways is special. Each one in its own. So you could. But then I was against sort of taking that further apart. So we could have had a split. The tyrosine and tryptophan there could have been a space between. But other than that, there could have been a space between these here. But anyway, so this is an attempt at sort of making some sense of biophysical features. Okay, observations. Don't kill each other. Who's going to be first?
Well, the functional residues, tryptophan and tyrosine, are obviously the lowest one for protein-protein interaction.
Be careful. So, so they are very special. Tryptophan and tyrosine are very special with respect to the data. But what did you exactly say?
Well, they have the lowest measure of.
So they are least expected. So they are underrepresented. which in some sense functional residues, tyrosine and tryptophan, and bind typically to substrates, small ligands. In that sense, that may be unexpected. They typically bind smaller molecules rather than bigger proteins. But that, yes, that would be observation number one. They stand out. Next observation. Nothing? But this is what you have to learn. You have to learn to look at your data and describe your data in a very neutral way, without interpretation. Just begin to see what you say. Say what you see.
Well, I have to say the interpretation of mine for some part is that it's strange because the arginine is sort of under-selected for protein and chain, but slightly selected in internal.
So you argue that you somehow did not expect this shape. Is that what you're saying?
Yeah, because it would have said. Basically, what I would expect is that I sort of.
Salt bridges are outside, not inside.
No, what I would sort of expect a spectrum where for chain. No wait, I misinterpreted something.
So again, let me repeat. What it means is that
Internally,
there are very
protein.
fewer of these are observed than expected. More of the arginines are observed in contacts, not argine, just argine as one of the binding residue, are observed more often than expected by chance in internal interactions, but only slightly more. Yeah?
How can actually residues be underrepresented in all three categories?
There's another observation. What I'm looking at is the subset of binding residues. And there clearly is an observation here that all are or that there is, this is a big statement here, but it's a very important one. All these binding interfaces differ from zero. So if we may remember quickly from the one we had previously, flexible, rigid, and something in between. And the in between was always zero. And that summed to 1. Here it doesn't at all, because the binding residues differ from all other residues. That is very clear, yeah? But but you already had your point. Is it a question?
It's
not statement. No, is it a statement or is it a question?
No,
kind of.
Question.
Good.
When you have considered the frequency in Swiss prot, did you only take those that
Yes.
are also in the?
Only those proteins.
Okay, and also only the one that is in the crystal has the crystal
Yes.
structure. Okay.
Yes.
Just to make sure that you don't run into the dark proteome issue.
I assume that we did. I'm not sure. I assume we did. Those days we didn't have a dark proteome idea. Those were the dark days before the dark proteome. Anyway, yeah?
So the chain-chain and protein-protein interaction seems to correlate, whereas
So blue and red...
in most of the cases, yeah.
So often blue and red are on the same side, I guess is what you mean, or are somehow more similar to each other than they are to green. Again, this is the kind of big statement. I really meant smaller statements, but this is a kind of big statement. In order to say that, you have to actually count, right? This is the other thing. So you have to sort of count how often, for instance, are they on different sides. 1, 2, 3, 4. 4 of 20 is the majority, right? This is exactly what this exercise is about. I'm not trying to say anything bad about you, but this is exactly. Thank you for doing this, because this is the exercise you have to go through when you put up with your result figure. There is so much we assume we see from a figure. And before we actually describe it in the method or in the paper, we are often not really clear what it meant. I have rewritten papers very often because of this step. because I made a statement, this means that. And I did exactly what you did now. And then I realized, wait a minute, if I count it, so visually it looks like much better correlated. But then, a simple way of counting, you may still be right if I computed the correlation coefficient. But when you do it in this way that I did, you see that sometimes counting is not quite giving you the same impression. And sometimes I rewrote the paper because then I realized I have to look at something else in order to make that correlation coefficient. And then I suddenly see something else. And in fact, I have changed the title and flipped the title in papers more than once. So it works, it doesn't work, it works, it doesn't work. In fact, let's not go through the number of iterations I did. But anyway, something else.
The amino acids in the first, second, and the fourth group are overrepresented for the internal interfaces.
And the others are, the hydrophobic are underrepresented, which in fact is quite surprising. True, because typically what you assume, hydrophobic residue is glue, and hydrophobic residues are the glue inside. Very surprising. True. Something else? Okay, let me give you a couple of others. So arginine and lysine, both are positively charged, right? It's surprising how much they differ. While, for instance, the DNA, the negative charges, agree much more with each other, arguably. I am surprised how, in fact, the agreement here between those sort of diverse class overall is. Then there are examples where we could pick the examples where we have the biggest difference. And the biggest difference would be, what would be the biggest difference between the two? That may in fact be the arginine here. It may be the isoleucine. Isoleucine and leucine, in fact, are similar. This is something that I expected. So, yeah?
Wouldn't we also expect more cysteine internal interfaces?
Yeah, so cysteine is a complication because cysteines bond to each other, but there are relatively few that form bonds. So the crucial cysteine, so first of all, cysteines are much enriched in the external proteins, secreted proteins. And the secreted proteins make up 20% of our genome, so 80% of the proteins have very few cysteines. It's just the secreted ones that are important for certain aspects, they have cysteines. They are short, and then yes, wait a minute, cysteine is here. They are overrepresented, right? You see them more than expected, although tiny. But that brings us to the next issue, and that's the issue. In some sense, I keep arguing the most important thing that you ought to bring home from my lecture is that you begin to network more, and this not Google more, but the second most important is ultimately asking for something that is missing in my graph. The question here is, they are different, right? That is clear. So the overall numbers are different. But are the differences meaningful? How would you find that out? And what is it that I'm missing? What is it that you should have prompted me to show you?
Significance.
Exactly. We need a statistical significance or something like that, right? And so typically we can do that for chi-square tests. The known problem is small data sets. So this is for medicine, right? You have 10 patients, and then it's too small. No problem here. We have millions of points. What is little less known is that for millions or many, many millions of very, very large data sets, is also a problem with the chi-square. But we have a significance of 10 minus 300. Okay? So this is very, very clear. These two distributions are different. And then the strange student has had a very... This is typically where you publish, right? And this stupid student, instead, what Yanai did is he said, well, okay, if I sampled proteins that start with A or B in the name, and I compared those to proteins that start with C or D in the name, how much would they be different? 10 minus 300. If I took proteins that were published by one author and compared them to proteins published by 2 authors, 10 minus 300. If I take random subsets, it was not 300, but it was still over 200, so it was still completely significant. Why? Well, so you go to the statistician, right, and you ask, what about your samples? Are they statistically independent? Well, we have proteins, right? And proteins evolved from some, no. Okay, so you cannot apply statistics. This, of course, is not, this is not the kind of thing that makes you happy, right? So from here on, no more statistics in anything that has to do with bioinformatics. We cannot live with that. We need it, right? So this cannot be it. There must be something else. Or is that really the issue? So we have to come up with a new test, ultimately. And the way we did it, what we call it find self, is a simple idea. You take the data set 1, 2, 3. So internal obligomers and external. And you take, say, 1,000 out of here, 1,000 out of here, 1,000 out of here, and then you take another 1,000. And you ask, these additional 1,000 by some Jameson-Shannon information content, are they most similar to this, most similar to that, or most similar to that? And if you always find them most similar to the class where it's picked from, then you statistically significantly can distinguish them. At least on the set of 1,000, right? So you repeat this test for 1,000 times, and what I'm going to show you on the table in the next slide is not three different types of interfaces, but 6 different types of interfaces. That's internal as we had it. Then there's domain-domain connections. Here is the protein-protein interfaces, and here I have what is called heteroobligomers. So I have 2 chains that I'm not entirely sure are from the same protein or not.